Welcome to our Think Like an Animal Scientist activity. My name is Emily, and we are so excited to have you here today for our Discovering the Scientist Within event. Before we get into the actual activity, we are going to hear from a current OSU student about a notable woman in the animal science field. So with that, I will hand you over to Ellie.
Thank you so much, Emily. As Emily has said, my name is Ellie, and I am currently a third-year student studying biochemistry and molecular biology here at Oregon State University. Even though I have a focus in medicine for my personal coursework, I do have an immense interest in animals, and so I'm really excited to share a notable woman in STEM who has contributed a great amount to animal science. And that woman is Dian Fossey. So Dian Fossey was born in 1932 in San Francisco, California. She developed a love for animals through riding horses from the age of 6. And she was originally told by her stepfather to pursue business, but she eventually turned her focus towards pre-vet focus and then eventually occupational therapy because she liked working with animals, but she also liked sort of like a humanistic aspect when it came to studying. So after she went to college, she decided, you know what, I want to travel the world. I want to see like what's out there. So she saved up all her money and took a trip to tour Africa. And that actually led her to pursue research with gorillas. And it's really cool because she was able to really get close to the community of gorillas within the mountains where she was studying. She imitated the behavior of gorillas in order to gain their trust. And she created such a close bond with them that she wrote Gorillas in the Mist. And that detailed her research, and that led her to advocate for conservation efforts. She did end up creating a conservation fund, and her message still lives on today. So with that, we hope that you keep Dian Fossey in mind as you continue thinking like animal scientists, and we hope that you enjoy that this activity brings you some perspective into thinking like an animal scientist. Take care.
Hi folks, welcome to Discovering Scientists Within program. This is the workshop called Think Like an Animal Scientist. My name is Dr. Kristen and I'm actually an animal scientist. So today we're going to walk through an activity together, and it is just like a real animal science experiment. So today we're going to really dive into how do animal scientists solve problems. So I will just preface this as saying, usually I teach this in person, and I'm the type of person that likes to do a lot of like questions and answering. And so I'm going to probably do that a lot today in this recording, is asking questions, and then I'll just give the answer as well. But I want to say something is that if you are following along in this video and you come up with an answer that's different than the one that I give you, that is totally okay. That's actually one of the things that I love about science, is that there's a lot of different ways that you can solve problems. So maybe you weren't exactly thinking the way that I was thinking. That doesn't mean that it was wrong. It just means that you had a different approach, and that's totally cool, and I love it. So let's just get started. So in all animal science experiments, or science experiments in general, you got to get prepared. You got to get your workspace set up and get it ready. So you had a lot of things in your kit as well as handouts, but the cool thing are these test tube racks. So it is these things right here. So there's 3 different pieces of plastic that are more see-through, and then there's 2 other pieces of plastic that are more opaque. So we're going to set up our test tube racks. Feel free to pause this video at any time if you need a little bit more time to assemble this. I've gotten pretty good at doing it myself, done this multiple times. So the way I like to do is make a sandwich. So I like to do the big holes on the top and the little holes on the bottom, so then the test tubes can kind of sit upright. And so there are different grooves. There's one side that has a groove, so you can slip this in. I wish I was like a really cool YouTube star. So like, look, I don't even know how people do it. There's a groove, and this groove fits on this. So you can kind of just guide it through like so. Be gentle with it. Don't have to force it. If it's getting a little stuck, that's totally okay. Sometimes it just needs a little wiggle and it will get on in there. If you try to force it too much, it could break. That's happened to me. It breaks, it's totally fine. I think that's why they give you actually an extra piece of plastic. Okay, so then here's a little bit of the tricky part. So as you can see, this is the top. I'm going to put the bottom piece in the bottom, so you kind of need to line it up on both grooves. Look at me. Told you I've had some practice with this. Kind of be patient and gentle with it, and it will fit in. Like so. So here's your test tube rack. These things are super handy and fun. And now get your test tubes out. So you will need the test tubes 0, 1, 3, 6, and 10. I filled mine up with water. If you fill it up to about the 10 to 12 millimeter mark, that's totally great. That's what we need. You will also have gloves in your kit. Feel free to wear gloves. What we're going to be working with today, there's a lot of really funky labels, and we'll talk about what's in this, but this is actually food coloring in water. It is a lot more concentrated, so it could stain your hands, and it may stain your workspace. So I actually have some paper towels listed, laid out right here that I'm going to put my test tube rack on. But wear the gloves if you don't want to stain your hands. I'm going to be bold today and not do it, but you know me. Final last words, right? And then, so you got your tubes 0, 1, 3, 6, and 10 on your test tube rack. You have one really, really dark colored tube that's got a really long acronym that we'll talk about later. And then you'll have another tube that will be a shade of a color, and it will have a name on it. So you need both of these out. And then you would need a plastic pipetter. Okay. So now that we are all set up for our experiment, now we can really do the fun part. So we're going to actually walk through the scientific method, and I will start this off by sharing my screen, and we will go through this together. So today I'm going to start this off by giving you a problem that we are going to solve together. So our problem that we're going to solve together is hyperthyroidism in cats is becoming much more common. Back in 1980, 1 in 300 cats was diagnosed with hyperthyroidism. Now it has risen to 1 in 10 cats. And the cause remains unknown. So this is the problem that we're going to solve together. So I said this to you. What are the first things that you think of giving you this problem? Well, if you're thinking to yourself, what is hyperthyroidism? How do we even get started? That's exactly what research is. And that's where a lot of scientists start out. So we're going to do some research together, and I'm going to give you different clues from different fields of literature. And this is what a lot of scientists do, is they actually read a lot of papers. And this is when really cool things happen, because you can draw connections from different things to help solve a real problem. So we're going to do this together. So if your first thought was, what is hyperthyroidism? That's a really great place to start. So let's start from the very beginning. The thyroid gland. The thyroid gland is a gland that produces hormones. Cats have it as well as humans. So as you can see in these pictures, where do you think the thyroid gland is in a cat? If you would say the throat, you are correct. And the interesting piece of this is that actually the placement and the function of a thyroid gland in cats is actually very similar to people. Cool, huh? So if a gland, the thyroid gland produces hormones, if it becomes hyperthyroidism, do you think it gets bigger or do you think it gets smaller? If you guess bigger, you are totally right. So if the gland gets bigger, do you think it produces more or less hormones? It produces more hormones. So on the bottom, this is different CT scans of cats. And on the bottom, which picture do you think is a cat with hyperthyroidism? If you guessed the huge one, you are correct. And it also probably is large too. That's actually very large. So when a cat gets diagnosed with hyperthyroidism, the gland gets bigger and it produces a lot more hormones. Now, you may be thinking to yourself, what happens when a cat gets hyperthyroidism? Well, this is a cartoon image drawing of a cat with hyperthyroidism. So this is some of the physical appearances that you can maybe notice in a cat. So they get very, very, very thin. And one of the interesting things that usually a lot of pet owners notice first is they are really hungry. They actually eat a lot, but they can't put weight on and they actually lose weight when they have hyperthyroidism. One of the reasons is because the thyroid hormones help regulate metabolism. So when it's out of whack, metabolism is out of whack too. And another interesting thing that usually pet owners notice first is their fur is very unkempt. So cats, especially when they don't feel well, they stop grooming themselves. And so that is some of the physical characteristics that usually people will see. And it's a good indicator that maybe they have hyperthyroidism. Okay, so we kind of talked about that. Now let's talk about how owning cats and having cats in our lives has actually changed in the US. And it's really interesting. So I have two different pie charts on this screen. One where cats live with owners in 2004 compared to 2014. So if you look at those two pie charts, what is one thing that you notice that's different? What's a trend that you're seeing? If you see that more cats are actually living indoors nowadays compared back to 2004, that is correct. So a lot of people are owning cats and they're bringing them inside. And there's a lot of different reasons for that. One reason could be they want to keep them safe. If you know where they are, you know where that they're safe, that they're not going to get lost or get injured if they're outside all the time. And another reason is some people say for ethical reasons. So for example, the Audubon Society is a group of people that are really passionate about wildlife and birds. And so a lot of people think that bringing cats inside is actually the ethical thing to do because they're not hunting birds. That's one reason. Another really interesting historical fact is this trend of bringing cats inside and living in our homes has actually been happening for quite a number of years, probably since the 70s. And that is from the invention of cat litter. So before the 70s, there was no such thing as cat litter. So people had cats and they just lived outside. And so that's kind of a really interesting thing is now having cat litter and getting it in grocery stores or pet stores. It's very common to us now, but back maybe in your grandparents' or great-grandparents' age, that wasn't a thing. Okay, so 2 clues so far. Third clue that I'm going to give you is a bit of a shift. So this one is a line graph that I'm showing you of the chemical usage trends in the US. So the number of chemicals that we come in contact or use in our everyday lives. So I want you to look at this green line right here, and it is being pointed by a big red arrow. So over time, what is happening with this, the amount of chemicals that we're using in our lives? If you think we're using a lot more of these chemicals, that is totally correct. Now, in science, we're very specific. So when I say the term chemical, that's a very, very broad and actually kind of a vague term. So let's get a little bit more specific. So I want to talk about one specific kind of group of chemicals, and those are flame retardant chemicals. So in this image, where do you think flame retardant chemicals are used? If you look at this picture and you're like, it's everywhere, Dr. Kristen, you're totally correct. It is in the insulation, the upholstery, even in the plastic casing, and in the carpet. So if this makes you a little alarmed, you shouldn't. It's actually kind of very normal. And I'll give you an example of why. I'll use a personal example. So when I was younger, I loved to go vacation at my grandmother's house. And so one of the things that I loved about my grandmother's house is that she had this really cool couch. It was actually made out of real wool fabric and wood legs. Nowadays, a lot of the furniture that we use in our house are actually not made from natural materials. I don't know if you saw my desk earlier. This is actually composite wood. So it's little pieces of wood that's pressed together and it's held together by actually plastic. And that is pretty common. So if you think about one of the more famous like furnishing stores, like IKEA, I love IKEA. So IKEA makes great furniture and it's really affordable. And one of the reasons why is they can mass produce this, and they can mass produce it because they're not using all natural materials. And so that really helps out with the manufacturing process. And one of the great things is that somebody can furnish their whole home for a really affordable price. You get couches, dining room tables, bed frames, all the things. It's a really great place to go shopping. I know I did it when I was in college. I furnished my whole college apartment that way. But let me go back to my example. So if a fire started in my grandmother's house, let's say way back when, it would take about 20 minutes if a fire started in the living room to completely engulf a house. Now, how long do you think, if a fire started in someone's living room on average in the US, how long do you think the fire will take to completely engulf a house? It's actually about 2 to 3 minutes. So it is great that we're using furniture that's very affordable, that can be mass produced, but one of the problems is that it can catch on fire pretty quickly. So for safety reasons, flame retardant chemicals are used in a lot of these things that we have in our house. So if there was a fire, that it actually prevents the fire from spreading so fast so we can get help and we can get out safely. So flame retardant chemicals are actually something that's very helpful in our society. Okay, but back to our problem. Let's think about this. So something that was really interesting that scientists saw was that this very specific chemical, Tdcpp, which is a flame retardant chemical, was found to alter thyroid hormone levels in fish, chickens, and humans. Okay, so now this is a piece of this puzzle. Maybe you're starting to see the image a little bit. One of the cool things that I love about science is how much it has changed in such a very, very short period of time, like within the last 5 years. So it used to be very, very expensive or time consuming or maybe a little bit painful to do some research. For example, back in 1980s, if you wanted to measure the amount of chemicals that a person gets in contact with a day, you would actually probably have to draw blood samples and draw a lot of blood samples. But nowadays, because of the inventions and the collaboration that's happening in science, there's a lot of different alternatives that are actually faster and less painful. For example, this wearable tech. So think about like your Apple Watch or Fitbit watches or things that can measure your steps or your heart rate. Those things are really becoming more commonplace now. And so there's this really cool wristband that people can wear that actually measures the amount of chemicals that we get in contact with. And actually one of them is the flame retardant chemicals again. So instead of doing a blood sample, people would just wear this wristband for, let's say, a week. Now bring it back to a scientist. The scientist will grind it down and extract the number of chemicals that they were in contact with. It just gets absorbed into this wristband. Pretty cool, huh? Okay, so I've given you all these pieces of clues. Let's go back to our problem and let's think about this. So why do you think cats are being diagnosed with hyperthyroidism now more than ever? Let's think of this of our hypothesis. And I'll also like to remind everybody again, there's a lot of different hypotheses that we can come up with to try to solve this problem. So the one I'm going to talk about is just one. So if you think about the number of chemicals that we use in our homes today, and especially those flame retardant chemicals that can alter thyroid hormones, and also cats are living more indoors. One hypothesis, if you think about all these different clues, is that cats are maybe being diagnosed and developing hyperthyroidism more nowadays because they're living indoors and they're laying on all our furniture. So if you think about like our couches, our beds, our carpets, so they're being exposed to more of these flame retardant chemicals. So that would be our hypothesis. Great. Now what? Now in science, what you have to do when you have a research question or a hypothesis is you usually have to compare two different things. So if we're thinking about cats that are living indoors now and they're becoming more exposed to flame retarding chemicals, and that maybe could be developing hyperthyroidism in cats, what would you compare an indoor cat to? If you thought about outdoor cats, it's actually a really great thing that we could do. So you can measure the number of chemicals that an outdoor cat is exposed to versus an indoor cat and see if these cats are being developing hyperthyroidism. So that's one really cool way to test our hypothesis. Now, one of the other things that everybody got in their kit is not only the handout, but everybody got a sample. So I mentioned earlier that this is just food coloring and water, but imagine that this is a sample from a cat. And so everybody has a different cat, and there is actually a handout that everybody got that has a picture of their cat and some information about them. So we will be using that to inform our data points that we're going to collect in a little bit. Okay, so we kind of already talked about experimental design. It's great to have a question, but you need a way to test it. And that is really just what experimental design is. But we're kind of missing one component. How will we measure the amount of chemicals that a cat is being exposed to? I kind of gave a bit of the clue when we're talking about the different research points. So we can get creative with this. And that's exactly what the researchers in this study did. Is very similar to a wristband that would absorb chemicals. The researchers in this study asked for the pet owners to put a very special silicone-based tag on their cats for a certain amount of time, so let's say a week, and that would absorb and be an accurate representation of the number of chemicals that cats are being exposed to. And they did this with indoor cats and outdoor cats. So it's actually a lot more of a humane way to measure something. You don't have to do blood samples. You don't have to bring them to a place that makes them really nervous. The cats are just in their normal environment, living their lives, doing whatever they normally do, and they just wear an extra little tag. Okay, folks, now time for the fun part. Let's start collecting some data and see if that can support or not support our hypothesis of cats that live indoors may be getting exposed to more flame retardant chemicals and also getting them diagnosed more with hyperthyroidism. So remember these handy dandy test tubes that I showed y'all earlier, and back to these. So everybody has a different test tube with a different name on it. This, as you can see, I have the cat's name is Cody. So this is a sample from the cat. So imagine a cat named Cody was wearing a pet tag collar for a week, brought the collar pet tag into the lab, and we were able to extract the number of chemicals that this cat was in contact with. And remember, this is only food coloring and water. This is not chemicals. This is just a mock experiment. So this is the cat sample that I have. And so I will share my screen. Everybody has a different sample. There's 4 different ones. And so check out the handout that has more information on the cat that you have. So some people may have the cat Sawyer. There's a picture, and the bottom there's some more information that the pet owners have provided. So does this cat have hyperthyroidism or not? How old is the cat? And where does the cat live and spend most of its time? So this is Cody. This is the cat that I have. So the cat does not have hyperthyroidism, but does have high levels of thyroid hormones. And cat's a little bit older and loves to sleep indoors. Some folks may have Pippin and Harley. So these are the 4 different samples in our study that we are going to measure the number of flame retardant chemicals that they came in contact with and see if that supports our hypothesis. So I will stop screen sharing that presentation. Okay, so we're going to collect some data, and I want you to measure the number of flame retardant chemicals that this cat has been exposed to. So how are we going to do that? Well, in science, we always compare things that we know to things that we don't know. So I'm asking you to analyze the concentration in your unknown sample or the sample from your cat. And I will tell you how to get started. So the other test tube that we have that is labeled Tdcpp, This is a concentration of the flame retardant chemicals. So we're going to make a standard curve off of this. So I am telling you that 1 drop of this equals 5 milligrams of the chemical, the Tdcpp. And so we're going to make a standard curve based off of that. So this is the really fun part. Remember, this can be a little bit messy. So if you have a nice workstation, I have paper towels over here. You can wear gloves or clothes that you don't want to get dirty. But if you are pretty good with a pipette, then you should be okay. So we're going to make a standard curve off of this sample. And so I'm going to help us out, and we're going to do this together as a group. We're also following the handout that has a little bit more of word text on the top of like the first paragraph. So we're also going through that. So to make a standard curve, we are going to put drops in each of these 5 blank tubes. So for tube number 0, how many drops do you think that we're going to put in? If you thought 0, you are correct. We're not doing any drops in this one. Now for tube 1, how many drops do you think we're going to put in here? You thought 1 drop, you are correct. So this is why having a nice test tube rack is easy. And in science, one thing I like to mention is we like to be very uniform. So if you're going to do different drops, try to be uniform in the number of size drops that you do. So I pulled up a nice amount of the Tdcpp, and I'm doing 1 drop in my test tube labeled 1. Put the cap back on, invert it. So you have a nice kind of a lighter shade of a color. So the next test tube is 3. And if you already were thinking ahead, we're going to do 3 drops in this one. Yep. And 6 in the other one. 6. And then test tube 10, we do 10 drops into this standard. Perfect. Okay. Now once you have put in all the drops, I inverted mine. Here's what mine look like. Do yours look similar? What are you noticing about these different standards? So for mine, the color gets darker the greater number of drops that we have. Okay, but now looking at these standard test tubes, we still don't know what the concentration of the unknown is. We're almost there with our standard curve. So we made it up, but we need to do a little bit of math. So remember I mentioned that 1 drop of the Tdcpp, I know the exact concentration of that, and that is 5 milligrams. So we can actually calculate that out. So we are going to be following along this handout. Okay. Now this is some of my favorite part of science, is doing a little bit of math. So for test tube 0, how many drops did we put in? We put in 0 drops. So it'd be 0 right here is where you would put this. So 0 times 5 is 0. And so the concentration of our test tube 0 is 0. Now, you may be thinking, why did you start with 0? Well, I'll show you in a little bit why 0 is very important mathematically. But another reason why a lot of scientists use zeros or blanks is what they call it, is in their standard curve, is because of a quality control. For example, if we were doing an experiment with DNA, we were extracting DNA, let's say from a hair sample from an animal. if we're making a standard curve of that DNA, if there was DNA that came up in the sample that nothing was supposed to be in it, that may tell me that my experiment may be contaminated. So DNA from another source, something that I'm not trying to study, maybe my own hair has gotten into the sample, or maybe accidentally like spilled something, that means that the experiment may be flawed and I have to start over. So that happens all the time. You actually learn a lot from things that don't work out in science, which I think is one of the coolest parts about it. So having a blank or a 0 in the standard curve is actually great for quality control. So you know that your experiment was, and you did your standard curve well. So for tube 1, let's look at that one. So how many drops did we put in? 1. So 1 times 5 would be 5. Now here's a little bit of the tricky part. We're doing a concentration, so that means we have to change some of the units. So we're actually multiplying by 10. So 1 times 5 is 5, and 5 times 10 is 50. So that is the concentration in this tube. Now, I want you to do the math for tube 3, 6, and 10. So fill out these 3 rows onto your handout. And you can definitely hit pause on this. So for 3, the concentration would be 150 concentration. What would this one be? 6, 300, and 10 would be the highest, and that is a 500 concentration. Okay, we're getting close, but still we have a little bit more work to do. So now we are going to graphically represent that standard curve onto a line. And I will show you why. It's pretty important in science. Okay, so I'm going to use the annotate function. So we are going to graph out this line that we just did. So for tube 0, we had 0 drops, so B0 is the standard, and also we had a 0 concentration, so you would put a point right here. Now for tube 1, so look on the x-axis, we go to 1, that's how many drops that we put in, and what was our concentration that we figured out? It was 50, so I'll put a point right here. Tube 3, so go over to the x-axis to 3, and then you would go up to the concentration to 150. 6 was 300, and 10 was all the way up to 500. Perfect. Okay, so now that we have plotted out our points from our standard curve, now we get to draw a line through these points. Oh, no. I guess I'm not the best at drawing a line. Yeah, that's pretty good, huh? Okay, so we have drawn a line of our standard curve. Now, a straight line is something that scientists love to see, especially when they're doing standard curve experiments, because mathematically that means that they are 99.9% confident that any guesses that they go off of this straight line, that they're pretty confident that could be the right answer. So this is great in a standard curve experiment because of a lot of different reasons. One reason is you don't have to do every single standard. For example, you only did 0, 1, 3, 6, and 10. I did not ask you to do 2, 4, 5, 7, 8, and 9. If I did ask you to do that, you probably are pretty confident that 2 would be around maybe 100, 4 would be maybe close to 200 of a concentration. So it does save in time. So that's one reason why having a straight line is makes a scientist feel very confident that they have a very sound experiment and they can use this standard curve to compare against things that they don't know. So that is one thing that's really handy with the standard curve experiment. And also, like we mentioned, the 0, starting from 0 is a good place, especially with the quality control. Okay, so going back to our initial problem, I wanted you to figure out the concentration in your unknown sample of the cat, right? We don't know what this concentration is, but now we do know what the concentration is in all of our samples of our standard curve. I know how many drops I put in each, and I know the concentration of each as well. So now the fun part is, now we like to see and compare. So what do you think my sample looks most like? You think it's 0, maybe 1, 3, 6, or 10? Let's see. Let me get you kind of like a better lighting over here. For me, I think my sample, Cody, looks a lot like three. Yeah, I think mine looks a lot like my standard 6. So what was the concentration in 6? Looking up on our line graph, that was 300. So if I think my sample looks most like my standard 6, and I know what the concentration in standard 6 is, I'm 99.9% confident that Cody's sample has a 300 concentration of Tdcpp chemicals, or those flame retardant chemicals. Awesome. So what about your sample? Did you have the same animal that I did? Maybe not. So for the people that had Harley or Pippin or Sawyer, so Harley was the very young animal that lives mostly outdoors, and that animal had a very low concentration of flame retardant chemicals at around 50. The animal that had the highest was Sawyer. That was the animal that did have hyperthyroidism, kind of looked very unkempt and very skinny in the picture. Also lived indoors and was a little bit older. So that was, Sawyer was about 500. So that was the highest. And then Cody, which is what I had, was about 300, was middle of the group at about 300. And then Pippin was even lower than that. Pippin was about 150 concentration. Oh, there's that straight line. Awesome. So actually what we did is something that scientists do all the time. It's a colorimetric assay. And so if you want to measure the concentration of something, you add reagents to it, and those reagents can react together to make a color, very similar to what we did with food coloring. So in this experiment, there's all these different wells or different samples. It mixes with the reagent. So some are darker, some are of a lighter color blue. And so what we did was something that we did by hand. We did the math by hand. But what this is really cool and automated is you would take that plate, you would have all these reagents mixed together, a color would form, but you put this plate inside of a plate reader. And really what it is, it just shoots light through each sample well. So each one of these little circles. And the darker the color, the less amount of light that passes through. And so then you can graphically measure it just like we did on that line graph. There is still a standard on here. So still a standard curve. So something that they know the concentration of each. And from there, you can do pretty much a mathematical equation to figure out the concentration of each sample. So what we did is very real experiment. I did colorimetric assays all the time. So talked about our findings, right? So our hypothesis originally was that we thought more cats were living indoors, so they're being exposed to maybe more flame retardant chemicals in our furniture, in our homes, and also that could be causing them to be diagnosed more with hyperthyroidism. That was our hypothesis originally. So what do we think? Do we think that our findings were valid or not? I will pull up the other presentation with the cat samples, and I told you the concentrations. So this animal, Sawyer, does have hyperthyroidism, does live indoors, is much older, and that one had the highest concentration, so that was 500, compared to the lowest was Harley. It is a very young cat, does not have hyperthyroidism, and was just recently rescued, so used to live mostly outside. So if you want to just think of those 2 points, do you think our hypothesis was supported? I think so. Now, the amazing thing about this is I mentioned this earlier. This is actually a real study. So this study was published in 2019, so not that long ago. So a group of scientists got together from toxicology and from the vet school at OSU, and they sought out to solve the same problem that we just did. They wanted to know why more cats were being diagnosed with hyperthyroidism. So they used these patented silicone tags, and they asked pet owners to put those tags on their cats, and they did the exact same thing that we did, is they measure the concentration of these flame retardant chemicals, and they saw very similar findings that we did. So really cool, huh? We kind of walked through an experiment. Now, the one thing that I do love about science is when you kind of solve one question, you can raise many, many other questions. So your mind may be racing at this point, like, wow, like all these cats are being diagnosed with hyperthyroidism. What can we do to help out cats? That could be something that we could explore more. Hence why you would do more research studies to try to help out cat health and try to do interventions. Some people may be thinking of, hey, you mentioned at the very beginning that cats and people both have thyroid glands that function very similarly. So is this happening to us too? Also a really interesting question that a lot of animal scientists are looking into right now. So that's why one of the reasons why I love animal science is not only do we go out to do experiments and we ask questions to help out animals, but a lot of these questions also help out people too. Right now, we don't know what's happening with people that are being exposed to these more chemicals that we're using in our everyday lives. These are things that we can be looking into. So all of these are really cool. This is one of the things I love about science, is that you just keep asking questions and you just keep learning and discovering new things. So I really, really hope that you enjoy this activity. Hey, these test tubes, you can always reuse them. You can dump them out. Make sure you clean up your workspace. All good scientists clean up after themselves and clean up after their workbenches. Enjoy the test tube racks. It's a really cool, fun thing that I get to share with y'all in the kits. And also, thank you so much for your time. I really enjoyed all of y'all. I hope you learned something cool, maybe had something new sparked interest in your own lives today. So without further ado, until next time, bye.